I'm Andrea Cline-Thomas. ISPOR focuses on key issues impacting healthcare. In this conversation, we're going to take a closer look at real-world evidence. We're going to begin this conversation with Nancy Dreyer, who discusses common data models and tokenization.
So a common data model is just like it sounds. We take data from different sources and try to make them speak a common language so we can combine them. It's essential for studying rare health effects and also for looking at safety issues where we need large populations. So having a common data model is great for that purpose. And tokenization is the new thing. Tokenization is an attempt to protect privacy through using a privacy preserving hash that takes generally a combination of private information you wouldn't want anyone to have. Your first name, your last name, your home address, a variety of your birth date, issues like that, and turns it into a number that can't be decrypted. And the beauty of the tokens is they're applied behind the firewall. So my institution, my healthcare institution, can use that token so that when they want to share their data, Nancy Dreyer is no longer identified by name, but by this hopefully unique token.
So how will these advances in real-world data enable the merging of large data sets at scale?
You asked about how this would allow us to merge large data sets at scale. And the beauty of the tokenization is that it allows us to match patients, for example, their pharmacy prescription fills. Do they actually go to the pharmacy and get the medication they were prescribed with an electronic health record where the doc said, I wish you would take XYZ, I think it will be good for you. So that basic merging using common data models and tokenization to link between different data sources really unlocks the goal to understanding how patients react to medications and who benefits and who is at risk of harm.
And what are some of the watch out areas?
Well, we are always worried about the accuracy of data linkage and tokenization. So the watch out areas are to think about, have you matched the right patients? And when things conflict, the big question is, what's the source of truth? How do we know what to rely on? Those of us who work in this field know that all real-world data are imperfect. It's not collected with the same care and attention that you would apply to clinical trials data, where you spend a fortune, so to speak, on each patient to make sure that every single data element is correct. When you want to study tens of thousands or millions of patients and you need answers, you can't afford to spend that much on each patient, but you need large data sets. So the value is in the large data sets. The worry is that you've got the right patients matched to each other so that you are in fact able to draw correct inferences.
So what are some important considerations as RWE enters this new era?
The new era of the availability of data for linkage and tokenization offers us so many opportunities. We need to start with the opportunities. For example, imagine taking a clinical trial and getting consent from patients to link to their long-term data. So we can find out, even with short-term data, were the benefits that we saw evident in surrogate outcomes in a clinical trial actually manifest in people's everyday lives. So does knowing that you changed some part of the pathway, does it make you feel better? Does it make you less symptomatic? So there are huge opportunities for understanding the next stage of clinical development. First we get from the test tube to the patient, and then we go from the patient in the highly controlled settings to the real world settings, where we have docs with various levels of experience, institutions with various levels of support. So we're opening up the potential to study more than just the mechanism of action of a drug or a device. So that's the good part. The bad part is, or the challenging part is, how do we make sense when things don't perfectly correspond? And I'm always telling my clinical development friends, you often have such situations, or you have encountered situations where clinical trials don't agree, but you trust those. But when real-world data doesn't agree, you immediately are suspicious. I think the challenge with real-world data is that it's a combination not just of the data, but of the methods that we do. The study design is critical. The analysis is critical. And then we have wonderful ways of testing the sensitivity of our results to various assumptions we made. That's where the gold is. Because if it's truth, it should show up in numerous populations, and your sensitivity analysis can tell you how much a result could be impacted by bias. Those are the questions I hope everybody keeps in mind.
We continue the conversation with Paul Aurora. The rare disease community is struggling with small sample sizes when conducting research using real-world data. How does your work in external control arms potentially help them?
Small sample sizes are a perennial challenge, both in rare disease and to some extent, many disease areas are becoming rare diseases because of the genetic and biomarkers involved in new drugs that are coming out. External control arms help with that because it reduces the burden on the patient population to participate in clinical trials where there may either be a reluctance to do so because of a lack of a concern about being randomized to a placebo or standard of care where they feel it won't work, or constraints around running clinical trials where you can't accrue substantial numbers. So external control arms potentially provide an avenue around that by leveraging existing data and reusing the same data and being more efficient with it. So you get to reuse the same control arm data numerous times. That's better for the patient and better for the manufacturer as well. The challenge, I think, is that we don't want it to be the Wild West. A new method comes out and the people generating the external control arms are using their own sort of ad hoc approaches to how to select the data and how to conduct the analysis. And then the people responsible for the policy decision making and regulatory reviewers are using their own set of criteria. The two groups need to be able to talk to each other, and it ensures, I think, that the approaches and methods are transparent and consistent, and that those that are responsible for making decisions based on that data are doing so on a standardized set of analytics. So I think it's very, very important that we're all working off the same spec sheet. One area of rapidly developing work is around synthetic data. Sometimes it's difficult to get access to real-world data to form external control arms, or it's difficult once you have access to the data in an academic setting, particularly to transfer that data across borders. Synthetic data is a new area of artificial intelligence that is helping solve both of those problems while at the same time addressing issues of patient privacy and consent. So it's a rapidly developing area. I think it's very early days, 5 to 10 years, I think, before it starts to become incorporated in decision making. But I think that's an area I would definitely keep my eye on, both on an academic and industry commercial setting.
Thanks for joining us. I'm Andrea Cline-Thomas. For more key topics about issues in healthcare, go to our website, ISPOR.org.